This presentation will describe biological exchange surfaces and is targeted at A-level students. The slide content has been specifically related to the specifications of the OCR exam board for A-level Biology A, but the information presented will be equally useful to any A-level student. As well as understanding the need for specialised exchange surfaces, you need to relate this need to surface area to volume ratio, metabolic activity, and single-cell versus multicellular organisms. An exchange surface is a part of an organism specialised to transfer molecules from one side to another. For example, living cells need to take in oxygen and release carbon dioxide. As the size of an organism increases, the ratio between the surface area and volume decreases, meaning that larger organisms can't rely on diffusion to transfer external molecules to all their cells because there isn't insufficient surface area for the amount of diffusion required by the many cells. Larger organisms also require more energy to move around, so the metabolic demand is higher. And in larger organisms, diffusion alone cannot penetrate to the centre of the organism. To calculate the surface area to volume ratio of a simple sphere, the surface area is determined by the equation 4πr squared and the volume by 4/3 πr cubed. Thus, as the radius of a sphere increases, the volume rises more rapidly than the surface area, leaving larger spheres with a lower surface area to volume ratio. The main features of an efficient exchange surface are large surface area, thin exchange surface, and good blood supply and ventilation. A large surface area is often achieved by folding or extending membranes, for example the root hair cell. The larger the exchange surface area, the more molecules can be exchanged at one time. A thin surface reduces the diffusion distance for the molecule, and therefore it increases the speed of exchange. An efficient exchange surface may be just one cell thick, as seen in alveoli of the lung. A good blood supply and air supply to a gas exchange surface maintains a steep diffusion gradient and ensures that the exchange molecules are constantly moving to areas where they're needed. The concentration of the molecule required by an organism is kept at a low concentration in the blood and a high concentration in the air at the exchange surface. In mammals, the gaseous exchange surface is found in the alveoli of the lungs. Airways in the lungs need to be large, strong, and flexible. Air passes through the mouth and down the trachea, which divides to the two bronchi, and the structure of the trachea and bronchi is supported by C-shaped rings of cartilage, preventing airway collapse when air pressure is low. Each bronchus further divides into bronchioles, which have walls of smooth muscle, allowing involuntary contraction to restrict airflow and elastic fibres to dilate the airway by recoil. The inner lining of the trachea, bronchi and bronchioles is composed mainly of ciliated columnar epithelium and goblet cells. Goblet cells secrete mucus to trap air particles and bacteria, and this mucus is swept upwards to the back of the throat by the cilia to reduce the risk of infection. Bronchioles terminate with clusters of alveoli, balloon-shaped structures specialised for gas exchange. Alveoli walls are 1 cell thick and composed mainly of squamous or pavement epithelium. They are lined with a surfactant to maintain their shape and surface function. And the lungs are protected by the rib bones, which are also involved in ventilation. Ventilation of the mammalian lung is achieved by the synchronous contraction of the diaphragm and intercostal muscles, drawing the diaphragm down and lifting the ribs up, causing expansion of the lungs and reducing internal air pressure, which then draws in air. Air is expelled from the lung by the synchronous relaxation of these muscles, which cause the diaphragm to arch upwards and the ribs to move in and down, increasing air pressure in the lung and forcing air out. The internal and external intercostal muscles oppose each other to move the ribs for lung ventilation. At rest, the external intercostal muscles connecting the ribs to the spine are relaxed, and the internal intercostal muscles connecting the ribs to each other are contracted. During inhalation, the external intercostals contract to lift the ribs, and the internal intercostals relax to allow the ribs to separate. It's important to note that the smooth muscle of the airways, specifically the bronchiole wall, is not involved in ventilation, Instead, this muscle is involved in involuntary constriction and dilation of the airway to restrict airflow when harmful substances are present in the air. Excessive contraction of this muscle occurs in asthmatic patients. Ventilation can be measured by vital capacity, breathing rate, and tidal volume. Vital capacity is a measurement of the maximum volume of the lung. The lungs of an average person have a vital capacity of 5 decimeters cubed, and this can be increased by regular exercise. Breathing rate measures the number of breaths taken per minute and is easily measured with a timer. Tidal volume is the volume of air taken in by the lungs at rest. The lungs of an average person have a tidal volume of 0.5 decimeters cubed. Gas exchange is measured by the volume of oxygen uptake over time. A data logger can be used to record secondary data, i.e. data collected by others, regarding ventilation, and a spirometer can be used to record primary data, i.e. data collected by yourself, regarding ventilation and gas exchange. To use a spirometer, a subject breathes through a mouthpiece connected to an oxygen chamber in a floating drum. The volume of air in the oxygen chamber changes with breathing, affecting the buoyancy of the drum, which can be recorded on a chart fixed to a second drum or electronically. The chamber contains soda lime to absorb exhaled carbon dioxide. The volume of carbon dioxide exhaled can be calculated and is equal to oxygen uptake. Bony fish use gills at the back of their buccal cavity to absorb oxygen from water into their blood and release carbon dioxide. Each gill contains 2 rows of gill filaments attached to a gill arch. Gill filaments have folds of lamellae to increase their surface area. Capillary blood flow through these lamellae is arranged such that water moves across them in the opposite direction. This is called countercurrent flow and increases the diffusion rate. To ventilate the gills, bony fish coordinate the mouth, buccal chamber, and opercular valves. First, the opercular valve is closed and the mouth is open. The buccal cavity expands to draw in water. The mouth then closes, the buccal cavity contracts, and the opercular valve opens, forcing water out and across the gills. To understand ventilation and gas exchange in insects, students should be familiar with spiracles, trachea, thoracic and abdominal movement, and tracheal fluid, as well as the examination and drawing of an insect dissection. Insects have an open circulatory system, so there are no blood vessels. Instead, air is supplied to the cells of the body through the tracheal system. Spiracles on the outer surface of the insect are connected to trachea to link them to the tracheal system, and tracheoles branch off from this system to reach all cells of the body, allowing gas exchange by diffusion. Insects use body movements to ventilate the tracheal system, circulating the air. Larger insects can coordinate contractions of the wings and the thorax and the abdomen in order to move air around the tracheal system. The histology images I will show on this slide are taken using a light microscope from stained thin slices of mammals, fish, and insects. It's important to be able to recognise all of the parts of the gas exchange system in these organisms from histology slides because they often look quite different to diagrams drawn to explain them. In the first slide, a view of one of the bronchi is shown. Note the C-shaped cartilage as well as the diameter of the lumen in comparison to a blood vessel, which helps to distinguish this bronchus from a bronchiole. The next slide shows another view of the mammalian lung to illustrate the alveolar duct and alveoli. The fish gill histology section clearly identifies the rows of gill filaments emerging from the gill arch and the additional surface area provided by the lamellae. In the final slide, an insect spiracle can be seen, providing ventilation to the tracheal system via a trachea. I hope this has helped with your A-level study of biology. Please look out for my other videos on other biology modules.